All righty. We have a test in 2 days. This is your opportunity to ask any questions that you have about the material that's going to be on that lab or lecture to help you to be successful. All right, great question. So the corpus luteum on the histology slide, there's a couple ways to be able to identify that. Let's see what I have. So again, this is just one example, but again, one of the keys with the ovary, and obviously we'll get into this in much more depth when we get to the reproductive system, is that we have these follicles that contain the oocyte in it. And while the oocyte is still in the ovary, it is in this follicle. And these are the follicular cells, those granulosa cells that are producing estrogen. But this swells up, this ruptures and bursts, and when it does, it releases the egg. And when it releases the egg, it often looks very much like this big popped balloon, because it's basically ruptured and burst. There's actually some women who can actually sense, the name for it is a German term, middle spurts, where basically they can actually feel the rupturing of the follicle, the slight tear of the ovary that takes place when it ruptures. You can actually see the scarring of the ovary and get a sense of how old a woman is just by looking at the scarring on her ovary from the popping of these balloons. But these granulosa cells are not only ruptured by the luteinizing hormone that triggers ovulation, but it also triggers the transformation of these cells. So these cells expand and grow and enlarge and can become this huge, massive glandular structure. Notice here we have one of those mature follicles, and next to it is this massive, big, huge glandular structure that is the corpus luteum. So clearly we see a follicle, we are still in the ovary, but we see this massive glandular structure next to it, and that is the corpus luteum. All right? So both of these would be examples. This is a early stage of the corpus luteum, and this would be the later stage of the corpus luteum. Okay. Again, remember, thecal cells are the cells that form the outer layer around a follicle. So they're those, that's where we find the thecal cells. And remember, those are the ones that produce the androgens. So that's that. It looks like Jessica also had a question. So notice we talked about 11 disorders today. So basically, you need to obviously know the 11 disorders, know what type of disorder they are. We had some autoimmune disorders. We had some lymphatic disorders. We really didn't so much have an immunodeficiency, although you could kind of argue that HIV is an induced immunodeficiency. So it isn't necessarily someone is born that way, and that's actually, you know, it's an acquired immunodeficiency. And so, so I think that would probably fall in that category. So yeah, so we have all three types that we have talked about. So you need to know the diseases, you need to know what type of disease they are. You need to be able to recognize general symptoms of them, what those diseases are associated with, what they affect, you know, what vitiligo does, what elephantiasis does. You need to have a basic understanding of the physiological function of it, how it occurs. You don't necessarily need to know the exact cell or something along those lines, although if there is a cell like those RS cells in Hodgkin's lymphoma. That's probably something. And any other characteristics about them that are stand out or may be unique about them. These are things that you could be asked about on both the lab and lecture exam. So I guarantee you'll be responsible for this material on both the lab and the lecture exam. All right.
I have a question.
Yes.
Please explain the complete and incomplete antigens. It's about the pathogens antigens, or it's about T cells antigens? I got confused.
I'm sorry. You were a little quiet at the beginning of the question. Could you say it again?
I'm asking about the complete and incomplete antigens.
Okay.
It's about pathogen antigens or T cells antigens?
Neither. So I mean, there is. So again, antigens are. So when we talk about a complete antigen, we're talking about those proteins that are found on the surface of cells. And when we talk about them being complete or incomplete, a complete antigen, remember, basically has two characteristics to it, right? What are the two characteristics that you need for a complete antigen?
Immunogenicity and reactivity.
Okay, so what does immunogenicity mean?
When it can stimulate specific lymphocyte to divide, right?
Right, exactly. So basically, like we talked about, in the activation of a T cell, that activation of the T cell comes from it binding to that specific antigen. And in that way, it is able to cause it to divide, activate it so that it can hunt that particular antigen down, okay? Then reactive means that any cell that is expressing that, the T cell is able to bind to it and destroy it, or it triggers the release of antibodies to it, or it triggers the release of cytokines to it, or something along those lines. And that's that reactivity. Okay, so things that are harmful to us are things that typically are complete antigens or have complete antigens on them. The problem comes when we have incomplete antigens, or what we call haptens. These are typically just a little benign piece of protein that are very common. Things like dander or dust or pollens or things along those lines, or the proteins from a peanut. The problem with those is that they in and of themselves are not immunogenic. So they can't cause the T cells to divide. But what can happen is that, especially when they bind to one of our own proteins, our protein and that hapten together becomes reactive. And so what happens is it is capable of stimulating an immune response. And because of that, it can cause T cells to be activated, it can cause the production of antibodies, and it can cause problems. That's one of the issues with, again, I hate to use my daughter as an example all the time to this, but she has a severe peanut and tree nut allergy. And so the problem is that if she comes in contact with that peanut protein, that special peanut protein that she's allergic to, it can cause T cells to be reactive. And again, notice it doesn't cause them to divide. So the first time she gets exposed to it, it may not have a severe reaction. But because of the memory cells that can be produced from that reaction to it, then the second, the third, the 10th time, after having 9 times of minor reactions to it, that 10th time may trigger a severe enough reaction where she undergoes anaphylaxis. So typically, incomplete antigens are things that are not dangerous by themselves, but typically when they bind to one of our proteins, they become reactive, and that causes an inappropriate immune response.
Okay, got it. Thank you.
Yep. What's next? Give me one quick second. All right, sorry. Nope, I see 2 new messages. Hold on, for some reason it's not scrolling. There we go. Great question. It is possible, and in some cases not uncommon for people to grow out of their allergies. This is especially true with some allergies like milk and strawberries. Again, I know most about nut allergies because that's what we deal with. People who tend to have fewer antibodies to them, under 10, whatever the units are of that, in some cases have as much as a 50% chance of outgrowing them. My daughter's values are well over 100, so she's not likely to grow out of it at this point. For whatever reason, the body becomes more tolerant of that incomplete antigen. In fact, that's actually one of the methods that are currently being studied and used, where we make the body tolerant. There were studies that started originally in Duke. Stanford is doing some of them right now, where the ones at Duke started with, you got like 1/64th, just like a shaving of a peanut, and they would be exposed to that and would eat it in a controlled environment where they were being monitored. And over the course of weeks, they would be added more and more of that protein to them, to the point where they could eat 10, 12 peanuts at a sitting without having a reaction to it. One of the interesting things they found in the original studies of that is that that tolerance wasn't necessarily long lasting. So what happened is, if the person stopped eating, they could sit down and eat 10 peanuts and would not have any reaction at all. Then they waited a month and tried a peanut again, and they had a reaction to it. So they went from not being able to eat peanuts at all to basically being forced to eat peanuts every day so they wouldn't have a reaction. They now have patches that can now present a small amount of the protein to try to build up that tolerance. And so they believe that most of that growing out of it has to do with building up a tolerance to that antigen that can occur. I mean, our immune response is dynamic that way. For many of you that live in the Central Valley, live here in Sacramento, you've probably experienced the opposite of that. Many people who move to this area never have any problems with seasonal allergies. And because this is a very agricultural dense area, after being in the Sacramento area for 2 to 3 years, many people who didn't have seasonal allergies before, become less tolerant to them and get seasonal allergies as a result. All right. Questions on that? All right, Kylene's got a question first, so let me answer that question first. Show the spleen histology. Yep, let's take a look and see what I have. Hold on. So mostly I have the slides that I used in class. Oh, I know I'm not finding that because we looked at that. I'm looking at my endocrine lecture. That's not in the endocrine lecture. Let's look someplace different. So again, these are not always necessarily the bestest of examples, but these are the ones that were either in your textbook or other places like this. So here is the spleen. Again, one of the keys, as we've talked about, is a lymphatic nodule is a lymphatic nodule is a lymphatic nodule. So they look kind of all the same. So there's things we need to look for that are going to help us to be able to, great question. There's a couple more, I'll come back to this, but let's answer this question quickly first. To look at things that are in the surrounding for them. Notice here we have a fibrous capsule, so we are definitely looking at some type of organ. But notice these lymphatic follicles are not all clustered together like they would be in a lymph node. Right, in a lymph node, all of them are located in the cortex, and then the medulla is completely clear, where these are spread out throughout the tissue, giving us two types of tissues. Now, notice when we look at this one, this one does look fairly red and this one doesn't quite look red. But notice when we switch to this view, right, we can tell the difference between the structures. And let's highlight it. Here is one of the follicles. Here is one of the follicles. Here is one of the follicles. So this white and red pulp isn't necessarily always the case. In this case, they've used a stain to emphasize the cells on this one, and it's given the color. They've gotten their name from what this tissue looks like when it is unstained. When it is unstained, it looks more like this because the red pulp, its job is to basically pull materials out of the blood, primarily dead and damaged red blood cells. So it stores a tremendous amount of hemoglobin in that area. So when it's not processed with stain, it tends to have a fairly reddish color. The white pulp, or what is known as the splenic nodules, are, as we know, clusters of lymphocytes. So they don't hold on to the hemoglobin. They don't have that iron in them, and so they're not going to have a reddish coloration to them. Now, while they primarily look like any other lymphatic follicle, the difference between these and the other ones is their job is to filter the blood. So if we need to filter blood, then that means there has to be a blood vessel associated with them. Here I would say is probably the most obvious example of one. What color can I use for this? Let's use green. Because it's a nice cross section, pardon me, not cross section, a longitudinal section through a blood vessel right through the center. But we can also very clearly see the cross section of a blood vessel at the center of that splenic nodule as well. If we switch to that other view with the different stain, right, again, it can be a little bit more challenging to see, but if you look closely, you can see indeed that there is a blood vessel right here that is passing through it. And I don't really see any other good blood vessels in this one. So I'll always use what I hope will be an obvious example of a blood vessel if I'm trying to give you a central artery through it. Here it is here. You can see that one there. In this one, it doesn't look like we see it in this plane of section. But there are plenty of obvious examples of those. So if I'm going to give you a central artery, I will give you something with an obvious example in it. But that's how you're going to distinguish the spleen. All right. Questions on this one? All right. Hold on. Okay, perfect. So let me go through these in order. No, again, you need to know that interleukins are specific examples of the cytokines, but you don't need to know the functions of all of the individual interleukins. I know you'll get that in microbiology, so I'm not going to bother going over it. I'll let your microbiology instructor deal with that. Great question. Genetics appears to play a role in hypersensitivity, things like allergies, but if someone in your family has a food allergy, it is more likely that you will have that you could have a food allergy, or that they tend to run in families. It appears to be more of a predisposition though than an actual thing for it. There tends to appear to be a lot of environmental factors. There are other interesting correlations between things like they were talking about today. Psoriasis can sometimes be associated with it, things along those lines. But it isn't the primary indicator. Nobody in both me or my wife's family on either side had allergies before my daughter had her allergy. And then after we found out about that, a year later, my sister found out her son has a nut allergy as well. So we went from a family on both sides, nobody with any allergies, to suddenly two. So, and again, there's a lot of interesting and curious research as to why there's been an increase in this in recent years. And there's so much of the information that is counterindicative, right? So one year they say don't give them nuts for the first 2 years. Now they're saying you have to give them nuts for the first 2 years. I mean, it's just there's a lot of time, a lot of research, but there's a big lack of understanding of how these things work. What's next? Thymus histology. Probably having a hard time with the thymus histology because you're not responsible for the thymus histologically. Right? Do you mean the thyroid gland? Notice your thymus is not on your list of things that you're responsible for histologically because there isn't anything super distinct about it, and so I'm not interested in holding you responsible for that. Remember, you are only responsible for the things that are on your histology lists. Now, again, I'm always good with knowledge for knowledge's sake, so if you're interested in studying that, I'm certainly not going to say no, but I will tell you that the thymus is not going to be on the exam histologically. Do you need to know what the thymus does, what occurs there, and all that kind of stuff? Absolutely. But I'm not going to show you a histology slide of it. I think it's on that list. So I think at the end of that list, isn't that where at the end of that, and then the lymphatic has a histology list as well. So both the endocrine and the histology both have histology lists, and that is the limit of what you're responsible for on those. For those lists. But of course, like I said, anything we went over, and I think someone mentioned this during one of my office hours, the thecal cells, for instance, are, I think, not on your histology list for the ovary, or they are, I don't remember what it was, but, thecal cells aren't on the handout, but obviously the thecal cells and those androgens that are produced by them are definitely something that you need to know on that. I think that's where they're, I think they're below the list. Here, hold on. Let's take a look. Oops. Okay, I don't want to minimize that. I just want it out of my way for a second. Because I have 85 windows open and I need to get to my desktop. So here's this handout, and yeah, so underneath the chart that we filled out is all the histology. The pituitary gland, the thyroid gland, parathyroid, pancreas, adrenal cortex, adrenal medulla, testes, and ovaries. And so yeah, so excellent. So thecal cells are here and you need to know what they do. But obviously over here for the ovary, we didn't mention the androgens here, because the androgens that are produced here aren't necessarily circulating hormones that have a specific effect. Instead, they are the building blocks that the granulosa cells use to make the estrogens. So that's why it wasn't listed here. So let's make this simple. I guess the answer to that would be yes. Am I likely to ask you to list all of them? Not on a lab exam, maybe in a lecture essay question type, but think of it this way. Do you need to know all the hormones that are produced by the anterior pituitary? Yeah, absolutely. And I would say, with the exception of melanocyte stimulating hormone, all the rest of the hormones of the anterior pituitary we know are controlled hormonally from the hypothalamus. So that means every single one of these 6 have to have a inhibiting and a releasing hormone from the hypothalamus to control it. So yeah, so yes. So we have it right here. So obviously, if I show you a histology slide, the things you need to be able to identify, whoops, that's what I want, would be obviously what the gland is, right? You need to know the hormones that are produced. Sometimes it's just the hormone. Sometimes it is a class of hormones. And if it is a class of hormones, you are also going to be responsible for the specific examples of them as well. Let me finish. I'm sorry?
So the class of hormones is releasing or inhibiting?
Yeah, both of these are classes. And then one specific example would be releasing thyroid stimulating hormone. Another specific example would be inhibiting thyroid stimulating hormone, right? Now let's take an easier example, right? How about if we scroll down to the cortex? Here in the cortex, the class would be glucocorticoids. The most common specific one would be cortisol, right?
Mm-hmm.
So yeah, so you do need to know gland, classes, specific examples, and then you also, oops, that didn't work. You need to know the targets of the hormone, the specific function of the hormone, and how the hormone is regulated. And then obviously, if it's histological, if I show you, for instance, right, a parathyroid gland, do you need to be able to recognize the specific cell, like a chief cell in anoxyphil? Or the acinar cells, right? I said you don't have to be able to recognize alpha and beta cells histologically. I'm not going to hold you for that, but do you need to know the zones, for instance, on the cortex of the cortex of the adrenal gland and things like that? Yep, absolutely. Hold on, let me go back. So those were those. Yeah, probably around 50% endocrine and 50% lymphatic. And so definitely, I would say definitely the lecture exam is going to be split pretty evenly between lymphatic / immune response and endocrine. So that would be the case. The lab exam, there might be a little bit more anatomy on the endocrine with all the histology. Then on the lymphatic, although there was a fair amount, so maybe that might be closer to, I don't know, 55-45, 60-40, something like that, but it'll still be pretty close. I mean, if you think about the amount of time we spent about a third of the time on lymphatic, on immune response, and on the endocrine, 1/3, 1/3, 1/3. So, but I think lymphatic tends to be a little bit smaller. So maybe 40, 40, and 10, or 45, 45, 10 for the lymphatic, something along those lines. I don't know. I guess there's a fair amount of anatomy. So it'll be close. Well, what specific histology do you want to see?
For example, the parathyroid gland, the pancreas, adrenal gland, so you can see like what they look like, or at least if you know a place where they have good pictures, maybe in their histology book.
So here's what I will tell you. In the lecture, when we were going through the lecture, I showed you specific examples of all of these. So you have all of the endocrine there, the histology. But remember, you've got your practice anatomy lab that has some histology, and that'll have decent histology for the endocrine. Your textbook, your lab manual, absolutely. Your histology atlas is going to be a great resource for this, where you'll have some great examples of all those things that'll show really nicely. And then as you get comfortable looking at those things, then I strongly encourage you to go to Google and start seeing lots of different examples. Because remember, my goal for you in this class isn't to memorize the one picture of a gland that I've showed you. That's not the goal. The goal is for you guys to comprehend and understand this information. And so the way you do that isn't just by memorizing one picture, it's by being able to recognize these particular components, what a pancreatic islet is, right? Or what the zona glomerulosa looks like, so that if I give you any obvious example of these things, that you should be able to discern what it is, understand what it is, and answer the question correctly. Mm-hmm. And again, the other huge advantage that you're getting on this exam that you don't get in the classroom is in the classroom, we go one question at a time, we go through it in order once, and then you get a brief review, all right? This time you've got all the pictures at once. So it allows you to compare pictures, to go back and forth and see, all right, how does this relate to this one there? And that can kind of help you sometimes to distinguish. I'm not sure if this is the thyroid or if this is the adrenal gland. Well, you can look at the two pictures or find a picture that you know was one and then compare it to the other one to try to help you with that. So that's going to be a huge advantage to you on this exam.
Mm-hmm. Mm-hmm.
Okay. So you need to know which hormones are amino acid based and which ones are lipid based. In that you need to know which glands produce which type of hormones. So what are the glands that we said that make lipid based hormones? Or glands or organs? that make lipid-based hormones? Anyone? All right, the cortex of the adrenal gland, perfect. What else? I can think of two more good ones. Or three, depending on how you think of it. Testes and ovaries. There we go, excellent. And then remember, we also said the kidney. Those are the organs that produce lipid-based hormones. All right, so do you need to know what hormones are produced by those four locations? Yep, so then it looks like you need to know which ones are lipid-based. Now, do you need to know which ones are amino acid derived and which ones are peptide? No, of course not. Right, but since you need to know which organs make lipid-based hormones, then you should know which ones are lipid-based and everything else is amino acid. So it's easier than you think. Any other questions? Great question. The pituicytes do not produce hormones. Remember, the posterior pituitary is the only endocrine organ that doesn't produce its own hormones. So it gets the hormones from the hypothalamus and stores and releases them. So yes, they store and release hormones, but they don't make their own. The diseases we covered today are on this exam, right? If you think about it, we talked about our immune response, we talked about our lymphatic system. So these are all related to things that either involve weaknesses to our immune response, where our immune response attacks our cells, or disorders of our lymphatic system. So absolutely, they fall right into this exam, so that you are responsible for them on Thursday.
I wanted to ask, does the hypothalamus target both of the pituitaries?
Yes, the hypothalamus controls both the anterior and the posterior pituitary, but remember, how it controls them is different. How does it control the posterior pituitary?
Neural control of the hypothalamus.
Yeah, well, yes, it controls the posterior pituitary neurally. And how does it control the anterior pituitary?
Hormonally.
There you go. So,
but
yeah.
the target, the answer, that would be regulated by question, right? But the function and target would be different?
Well, so again, you're flipping the questions. If the question is, what does a releasing hormone target? The answer to that is the anterior pituitary. If I'm asking you how thyroid stimulating hormone in the pituitary is regulated, your answer to that is a releasing and inhibiting hormone from the hypothalamus. Right? So you're talking about two different hormones. Hormone 1 is the releasing hormone. What is its target? Right? Hormone 2 is thyroid stimulating hormone. And for that, we want to know how it's regulated. Now, obviously, the two are related because one regulates the other, right? Or one targets the other.
So inhibiting hormone would be thyroid stimulating.
No, there is an inhibiting thyroid stimulating hormone. The hypothalamus produces a specific hormone that is known as an inhibiting thyroid stimulating hormone. When it releases that hormone into the blood, it goes to the anterior pituitary, and it stops the anterior pituitary from, or at least slows down the anterior pituitary to make less and to release less thyroid stimulating hormone.
Okay, thank you.
Yep. Leptin also targets the hypothalamus. That is correct. Come on. There we go. All right. So again, one of the things we know about the hypophysis or pituitary gland is that it is made up of two different types of tissues with two distinctly different, well, I guess not different types of functions, but at least different hormones associated with them. We have this much lighter color region. What is this region?
Posterior pituitary. It's
Posterior pituitary.
okay.
And it is, exactly, it is nervous tissue. Excellent. Notice there is also a stalk of nervous tissue that connects it to the hypothalamus. What is that stalk of nervous tissue called?
Infundibulum.
Infundibulum. Excellent. Then over here, we have this entire structure over here, which, what is this entire structure here?
Adenohypophysis.
Adenohypophysis, or the anterior pituitary. Excellent. And what type of tissue is it?
Glandular.
Glandular tissue. Excellent. This one is much larger, and we can kind of divide this one up into three regions. Right? So if we cheat, right, so we have this part up here, we have this part down here, and we have all the rest. This part up here associated with the infundibulum, what is it called? Pars tuberalis. Excellent. This is the pars tuberalis. This part that is adjacent to the posterior pituitary is the pars intermedia. And then, as someone mentioned, the bulk of it is the part that's furthest away called the pars distalis. Excellent. All right. Now, the pars intermedia is histologically distinctly different from the rest of the anterior pituitary. All the rest of the anterior pituitary has different clusters of cells that are capable of producing all of the hormones of the anterior pituitary except one. What again is that one hormone that can be produced but isn't typically produced here in the pars intermedia? Yep, although don't abbreviate on the exams, but absolutely. Melanocyte stimulating hormone. Excellent. Yeah, don't use abbreviations on the exam, remember. Excellent, that melanocyte stimulating hormone. And in fact, we actually looked at that when we looked at it histologically. Histologically, we can actually see that while right here, let's do it this way. This is the boundary right here between the posterior pituitary, that's not gonna work, posterior pituitary and the anterior pituitary. Here are those pituocytes that we talked about here. But there is this region in here of the glandular anterior pituitary. Remember, this is part of the anterior pituitary. This is still glandular tissue, but this is that kind of intermediate weird region, that pars intermedia, where we can produce the melanocyte stimulating hormone. And then all the rest of these. Now again, as I look at this one glandular cell right there, do I know exactly what hormone is being produced by that one? Do we? Do we know? Can we determine what hormone is being produced and released by this one cell here? No, we're not able to figure that out, but we know what 6 it could possibly be. Right now, with different stains, might it be possible to tell what they do? Sure, yeah. Do I expect you guys to be able to do that? No. All right. Questions on that? I think that's it that you're responsible for histologically for this, right? You guys have the list? Since no one's saying anything, I'll assume yes. All right. Any more questions? I think it is. I don't actually, you know what? I don't actually know that, but I believe calcitriol is, because it's cholesterol based. All right. Something bad's got to be going on because I have half my family calling me. Doing a quick search. Yeah, it looks like it is cholesterol based. So yes, it is. That's what I thought. It is indeed a lipid base because it comes from the kidney. Yep. I like it when one of the things that I say are accurate. That's always fun. All right. Any other questions? All right. Well, I will remind you, I have office hours tomorrow from 12:30 to 1:30, right after my lecture. And then, and then I will actually have office hours right before your exam as well. So I'll be available there. One of the fun things about this is I don't get to make the tests. I've put all the questions into the bank, and the random dice rolling gods determine whether or not your test is hard or not. So you have to talk to the randomizer on the on Proctorio to determine, or on Canvas to determine whether your class is hard or your test is hard or not. I unfortunately have no say. So I will root for all of you having easy tests. But like I said, you're on your second exam in 431. You guys know what you need to do to be successful in this. You know how to prepare for this. You've now all had a test from me, lab and lecture exam. You know exactly what to expect. The exact same way I made the last test is going to be the exact same way I make this test, so you know what is expected from you. That's not how the curve works. Nope. All right. Plus, good luck getting everyone to agree to that. All righty. Any other questions? There you go. Exactly. Time to offer a good sacrifice. There you go. All righty. Then you guys have a good evening. Be safe. Pay attention to the vice president debate that's going to go on tomorrow night. Should be interesting. Should be entertaining. So yes, make good decisions. Study hard. Be safe. Listen to the doctors, not to the politicians. Wear a mask. Yes, absolutely. Wear a mask. Make good decisions. All right. Take care, guys, and I will see you in a couple days.